Usually when we talk about a biological superpower, we picture things like, I don't know, a cheetah's speed or a bat's echolocation.
Right, yeah, the flashy stuff.
Exactly, the flashy stuff. But you possess a superpower that is arguably just as mind-blowing, and you probably haven't thought about it once today. Your skin completely replaces itself every few weeks.
It really is incredible when you stop to think about it.
It is. I want you to just imagine the immense logistical challenge of this. I mean, you have billions of cells. How do they know exactly when to divide, when to mature into a hardened protective barrier, and, when to call in the immune system if something goes wrong?
It's an absolute marvel of cellular logistics. Like if it were a factory, it would be the most complex, perfectly timed assembly line on Earth.
Yeah, the timing has to be perfect.
Exactly. A fraction of a millimeter off or a timing delay of even just a few hours, and the whole system crashes.
And when it crashes, we see the devastating results, right? Chronic wounds, severe eczema, psoriasis, or even skin cancer. Which brings us to the source material for today's deep dive.
Yes, a really phenomenal paper.
We are looking at a dense, incredibly cutting-edge 2026 paper published in the journal Science by Winge and colleagues. It's titled, let me get this right, Ubiquitin-like proteins NEDD8 and SUMO2 control epithelial homeostasis, regeneration, and inflammation.
It is a massive study. I mean, the researchers essentially threw the kitchen sink of modern molecular biology at this problem.
They really did.
Yeah, they used genomics to look at the DNA, proteomics to look at the proteins, and spatial transcriptomics to map it all out.
I want to stop you right there, actually, because spatial transcriptomics sounds like something from Star Trek. Are we just talking about mapping where specific genes are turned on in a physical slice of tissue?
That is a great way to put it, yeah. Instead of just, grinding up a piece of skin and looking at a soup of genetic material.
Which is what they used to do, right?
Right, exactly. But spatial transcriptomics lets you look at the tissue under a microscope and see exactly which cells are reading which genes in real time, like in their actual physical location.
Okay, let's unpack this. Our mission today is to explore what they've found with all these tools. We want to see how two microscopic protein tags act as the absolute master switches of your skin's fate.
The master switches, exactly.
We're going to unpack how they balance cell youth and maturity, what happens when they break down, and the frankly mind-blowing molecular mechanisms they use to control your RNA. So set the stage for us. What is actually happening in the epidermis on a normal Tuesday?
Okay, so the epidermis, which is the outer layer of your skin, it's what we call a stratified tissue. Think of it like a biological layer cake.
Oh, layer cake.
Okay.
Yeah.
So the cells at the very bottom, they sit on a foundation called the basement membrane that makes up the basal layer. And these are the progenitors.
Progenitors.
Right. Their whole job is to divide, multiply, and, keep the tissue populated.
They are the youth of the skin, basically, the fresh recruits.
Exactly.
Yeah.
But they can't stay young forever because space is limited, right? So as new cells are born at the bottom, the older ones get physically pushed upward.
Up toward the surface.
Up toward the surface. And as they move up, they undergo this radical transformation called terminal differentiation.
Terminal, meaning they stop dividing entirely.
They completely shut down their reproductive machinery. Instead, they mature, and they completely transform their internal structure to become a flattened, hardened shield.
Like armor.
Literally armor. They build it around themselves. It's called a cornified envelope. And then eventually they reach the surface, they die, and they flake off as dust.
So there is this constant conveyor belt. Divide at the bottom, harden at the top, flake off. Let's do a quick recap here. The big question is, how does a single cell know where it is on that conveyor belt? I mean, does it just guess based on how far it's been pushed?
That's exactly what Winge and the team wanted to find out.
Okay, so how did they look for the controllers?
They turned to a highly sophisticated technique called perturb-seq. So they took human skin cells, keratinocytes, and they used CRISPR gene editing to systematically knock out 202 different genes.
Wait, 202 genes?
Yeah, 202 genes that they suspected were involved in this conveyor belt process.
So they edit the gene, they break it, and then what? Do they just watch to see if the cell dies?
Well, that would be the old school way. But because it's perturb-seq, they use single-cell RNA sequencing to look at the exact genetic response of every single individual cell after the edit.
Oh, wow.
I know. They tracked over 8,500 RNA transcripts and more than 9,100 proteins across 10 different time points.
That is an absolute ocean of data. You're tracking thousands of moving parts across thousands of cells just to see what happens when you remove one specific gear.
It's massive. And out of all that noise, the data pointed a giant flashing neon sign at two specific proteins, NEDD8 and SUMO2.
NEDD8 and SUMO2. Okay. Now, the paper calls these ubiquitin-like proteins, or UBLs. If you know a little biology, you might recognize ubiquitin. It's famous for being the cellular kiss of death, right? Like a tag that marks a protein to be thrown into the cellular trash can, the proteasome.
Yep, the proteasome.
But surely NEDD8 and SUMO2 aren't trash tags, right? Because if they are, how on earth is a trash disposal system controlling tissue regeneration?
That is the crucial distinction here. While they are structurally similar to ubiquitin, I mean, they look like cousins, they are doing something completely different.
Okay.
They aren't marking proteins for destruction in the proteasome at all. They are physically changing how those target proteins function. And the data showed that NEDD8 and SUMO2 are pulling the cell in completely opposite directions.
So it's a tug of war.
A very precise tug of war. So NEDD8 is the tag that sustains the progenitor state.
Okay.
When it is attached to its targets, it keeps the cells dividing, regenerating, and essentially young.
So NEDD8 is basically the Peter Pan tag. It refuses to let the cell grow up.
That's a perfect analogy. And SUMO2, on the other hand, enables terminal differentiation. It tells the cells to stop dividing, mature, and form that hard barrier.
Making SUMO2 the career counselor tag.
Exactly.
It pushes the cell to grow up, stop messing around, and get to work, building the skin barrier. So, quick summary for everyone listening. We've got the Peter Pan tag, NEDD8, keeping things young and dividing, and the career counselor tag, SUMO2, telling things to harden up.
Right.
But if we have this delicate balance, what happens if we tip the scales? Like we drug the cells to stop the Peter Pan tag from working?
The researchers used specific pharmacological drugs to test exactly that. They used a drug called pavonetistat to block NEDD8, and another called subasumstat to block SUMO2.
And the results?
When they blocked NEDD8, the cells immediately stopped dividing and prematurely differentiated. They just grew up way too fast.
Which would completely ruin the tissue because you run out of dividing cells at the bottom to keep the conveyor belt moving.
Precisely. And then they ran the reverse experiment. When they blocked SUMO2, the career counselor tag, the cells were trapped as endless progenitors.
Just dividing and dividing.
Yeah, they just kept dividing
and
refused to mature into barrier cells, which means you have no armor.
So they proved it in a Petri dish. But, you know, lab-grown cells are one thing. A dish isn't a breathing organism with an immune system and blood vessels. To see if breaking these tags actually causes the skin diseases we see in humans, they had to engineer mutant mice, didn't they?
They did. And they used a very elegant genetic tool called the CART14 crete system to do it.
CART14 crete. That's a mouthful.
It is. But without getting bogged down in the deep genetics, this essentially acts as a biological timer.
How so?
It allowed the scientists to breed mice that developed totally normally. But then, at a specific time of the researchers' choosing, they could administer a chemical trigger.
Okay.
And that trigger flips a genetic switch and deletes either the NEDD8 gene or the SUMO2 gene, but only in the animal skin.
That's wild. Okay, let's start with the NEDD8 knockout mice. You flip the switch, the Peter Pan tag is gone from the skin. What physically happens to the mouse?
It is dramatic. Because the cells lose their youthful anchor, they prematurely differentiate. The skin becomes visibly thickened, which is a condition called hyperplasia.
Hyperplasia.
And it becomes highly scaly, which is hyperkeratosis.
But visually looking thick doesn't necessarily mean it's actually working as a shield, right? Did they test the barrier function?
They did. They measured something called transepidermal water loss, or TEWL.
TEWL.
Yeah, it's a standard way to measure barrier integrity by seeing how much water evaporates right through the skin. And in these mice, the TEWL skyrocketed. The barrier was fundamentally broken, even though it looked really thick.
And I imagine a broken barrier is bad news if you get a cut.
Oh, the wound healing crisis was the most alarming part.
Yeah.
So the researchers gave the mice a full thickness wound. Now, in a normal, healthy mouse, a wound like this perfectly heals and re-epithelializes in about 14 days.
And without any NEDD8?
They completely failed to heal. 14 days later, the wound was still wide open.
Wow.
The cells just couldn't maintain the progenitor state needed to divide and crawl across the wound bed to close the gap. And to make matters worse, the tissue was swarming with massive amounts of neutrophils.
Neutrophils, right. Those are the first responder immune cells, the
Yes.
ones that rush in and cause that angry red inflammation.
Yes.
But why were they swarming a wound that wasn't healing?
Well, the loss of NEDD8 triggered the epithelial cells to release very specific distress signals. The paper highlights a chemical signaling pathway called the ANXA1 FPR12 axis.
Okay, ANXA1 FPR12 axis. Let's translate that. What does that actually mean in plain English?
Think of it like a cellular flare gun.
A flare gun. I like that.
When the skin cells realized their machinery was breaking down without any NEDD8, they fired off these ANXA1 proteins. Those are the flares.
Right.
And then the SPR1 and 2 receptors on the neutrophils saw those flares and came rushing in, completely flooding the area. It created this state of chronic rogue inflammation.
That sounds absolutely catastrophic. What about the SUMO2 knockout mice, though? Does losing the career counselor tag cause the same chaos?
It causes a completely different kind of chaos.
Okay.
Without SUMO2, the skin failed to differentiate properly. The barrier also broke down, so you still see that high water evaporation, the high TEWL.
Right, because they can't form the armor.
Exactly. But here is the striking contrast. When they wounded the SUMO2 knockout mice, they healed normally.
Wait, really? The barrier's broken, but they can still close a wound?
Yes, because they still had NEDD8. They still had the Peter Pan tag, which meant they had the progenitor capacity to divide and close the wound bed.
That is fascinating.
And interestingly, instead of an angry swarm of neutrophils, their skin was infiltrated by T cells.
So if you've ever known someone with a chronic diabetic foot ulcer that just won't close or, severe inflammatory psoriasis, you are looking at this exact molecular failure in real life. We're seeing the literal root of why skin fails to heal.
It really bridges the gap between the lab and human disease.
It does. Let's do another quick check-in. So far, we've got NEDD8 for youth and healing, SUMO2 for barrier maturity, and losing them completely wrecks the skin in different ways. But this brings up a massive lingering question for me.
What's that?
How are these tiny tags actually executing all these complex orders? I mean, how does attaching a tiny protein to another protein cause an entire tissue to change its identity from a dividing cell to a hardened shield?
What's fascinating here is that the researchers went looking for exactly that answer. They proved that NEDD8 is not just tossing differentiation proteins into the garbage. It is doing something much more sophisticated.
Okay.
It is controlling the lifespan of the cell's RNA.
RNA. Okay, the instruction manual is copied from our DNA that gets sent to the factory floor to tell the cell what proteins to build. You're saying NEDD8 is controlling how long those manuals last before they disintegrate.
Yes, exactly. And to prove this, they treated the cells with a drug called actinomycin D.
Actinomycin D.
Right. This drug is a hard stop for the cell. It completely freezes new transcription. The cell cannot make any new RNA.
So they freeze the factory, and then what?
By freezing the production of new RNA, the researchers could track the RNA that was already on the factory floor and see how long it survived before naturally degrading.
Oh, I see.
And they discovered that NEDD8 is the critical factor determining how long specific RNA manuals survive before being shredded.
But wait, NEDD8 is just a protein tag. It can't read or hold RNA itself, right? It must be tagging something else that actually handles the RNA.
Exactly. So they ran a genome-wide CRISPR suppressor screen combined with immunoprecipitation mass spectrometry to find the middleman.
Whoa, immunoprecipitation mass spectrometry. That sounds like a serious mouthful. Are they basically just using chemical magnets to fish out whatever the NEDD8 tag is physically stuck to?
This is exactly what they did. Immuno refers to using antibodies like magnets to pull out NEDD8, the precipitation part.
Right.
And then they used mass spectrometry to identify whatever came along for the ride.
Like weighing it to see what it is.
Yes. And they found the holy grail, an RNA-binding protein called HNRNPU.
HNRNPU. You know what? Let's call it the molecular librarian.
The librarian is a fantastic analogy. HNRNPU's job is to physically grab onto RNA strands and stabilize them so the cell's enzymes don't chew them up.
Okay, that makes sense.
Here's the mechanism they uncovered. When the librarian, HNRNPU, is tagged with NEDD8, it tightly grips and stabilizes the specific RNAs that keep the cell in a progenitor youthful state.
So NEDD8 is like the library card, or maybe a really strong cup of coffee for the librarian. As long as the librarian has it, they hold on to the youth blueprints.
Yes.
But what happens when you remove the NEDD8 tag?
When NEDD8 is removed, HNRNPU physically changes its shape and its grip. It drops those progenitor RNAs, leaving them to be shredded.
Wow.
And instead, it grabs and stabilizes RNAs that code for differentiation and maturity.
That is incredible. It's not that the cell stops reading the DNA in the nucleus. It's that NEDD8 acts as a master toggle switch on the factory floor.
Exactly, a toggle switch.
Tag is on, the librarian protects the youth blueprints. Tag is off, the librarian drops the youth blueprints and picks up the armor blueprints. It's deciding which manuals are saved and which are thrown in the incinerator.
It is a profound layer of regulation that we simply didn't fully appreciate before this paper. I mean, it explains why blocking NEDD8 forces the cell to mature so violently. The progenitor blueprints literally disintegrate before the cell can even read them.
So we've got the mechanism down, the toggle switch on the librarian. But here's where it gets really interesting. And I'll admit, a little confusing, so I need you to clarify this for me.
Sure, go ahead.
We talked earlier about how the NEDD8 knockout mice had that massive angry red neutrophil inflammation shooting up those cellular flare guns.
Right, the Angzia1 flares.
Yeah. But if NEDD8's main job is just acting as a toggle switch on the librarian to hold on to youthful RNA, what on earth is causing the immune system to panic? I mean, why does losing an RNA stabilizer cause massive inflammation?
That is the exact paradox the researchers needed to solve. And the answer involves a protein called NRF2. NRF2 is a well-known cellular stress alarm.
But why is the cell stressed in the first place?
Think about what happens when you force a cell to prematurely differentiate.
Okay.
The NEDD8 tag is gone, the librarian drops the youth blueprints, and suddenly the cell is forced to build a hardened barrier. But it doesn't have the resources or the proper setup yet. Its internal machinery starts breaking down under the strain.
Oh, like trying to build a brick wall before the cement has even arrived, things just start collapsing.
Perfect analogy. And when cellular machinery breaks down, it leaks reactive oxygen species. This causes severe oxidative stress.
Ah, okay.
That oxidative stress trips the NRF2 alarm wire, which triggers those protective but highly inflammatory pathways we talked about. The sudden loss of NEDD8 was causing structural collapse, which triggered the NRF2 panic response.
So the cell loses its Peter Pan tag, the RNA gets shredded, the cell is forced to differentiate before it's ready, its engines leak, and it basically hits the panic button.
Exactly. But, you know, in science, a plausible story isn't enough. You have to prove causation.
Right, of course.
So they created a double knockout mouse. They genetically engineered a mouse that was missing NEDD8 in its skin and missing the NRF2 alarm protein.
Oh, wow. So they broke the barrier, but they cut the wire to the alarm bell. What happened when they did that?
The result was beautiful in its clarity. The severe redness, the erythema, and those massive swarms of neutrophils, they completely vanished.
The inflammatory panic just stopped.
It stopped.
But I'm guessing the skin was still scaly and the barrier was still broken.
Yes, because the librarian was still dropping the blueprints. This brilliantly proved that the inflammation was a secondary distress alarm. It was the cell screaming for help because its machinery was failing.
I see.
But the fundamental structural defect, the failure to maintain youth and build a proper barrier, is entirely hardwired to the loss of NEDD8 and that RNA-binding protein, HNRNPU.
That distinction is so vital for medicine. If you just treat the inflammation in a chronic wound with steroids or something, you're really just turning off the alarm bell. You aren't fixing the fact that the factory is using the wrong blueprints.
Exactly. And this brings us to perhaps the darkest implication of this entire mechanism: cancer.
Right, because if NEDD is the fountain of cellular youth, keeping cells dividing, cancer is essentially a disease of uncontrolled cellular youth and division.
Exactly. The researchers looked at 41 samples of spontaneous human epidermal cancers, specifically squamous cell carcinomas.
Okay.
And when they analyzed the proteins in these tumors compared to healthy skin, they found massive, highly elevated levels of NEDD8 tagged HNRNPU.
So the cancer cells are intentionally hijacking this exact pathway.
Yes.
They are hyperactivating the Peter Pan tag to ensure the librarian never ever drops the progenitor blueprints. They stay perpetually young, they constantly divide, and they utterly refuse to mature into harmless barrier cells that would eventually flake off.
They are exploiting the very mechanism of tissue regeneration to fuel their own endless growth. The toggle switch is just jammed in the on position, and the cell just divides and divides.
It's terrifying, but also incredibly illuminating to see the mechanics of it so clearly. You know, let's take a step back and recap this empire of the skin. We've covered a lot of molecular ground today.
We really have.
Basically, we have these two tiny protein tags, NEDD8 and SUMO2, and they run the show. NEDD8 is the anchor of youth and regeneration. It tags the HNRNPU librarian, stabilizing the RNA blueprints that keep cells dividing and capable of crawling across a wound to heal it.
Right.
And then SUMO2 is the driver of maturity, forcing cells to build the protective barrier that keeps our water in and the outside world out. Losing either one results in disaster.
Yeah.
broken barriers, failed wound healing, and rogue inflammation from leaking cellular machinery.
If we connect this to the bigger picture, this raises an important question for you to consider. Right now, pharmaceutical companies possess UBL inhibitors, like the pivenotastat used in this study. But they are primarily used, or being tested, as absolute sledgehammers to treat cancer.
Yeah.
By trying to shut down that endless division systemically.
Right, whole body.
Yeah. But what if we think smaller and more targeted? What if we think about the skin barrier itself?
You mean if our cells are constantly using these tags to read the room, to decide whether to heal, to mature, or to sound the inflammation alarm? Could we hack that system locally?
Could we eventually develop topical creams or targeted therapies that precisely tweak the levels of NEDD8 and SUMO2? Imagine a treatment for a chronic diabetic foot ulcer that doesn't just try to keep the wound clean, but actually applies a localized burst of a NEDD8 pathway activator.
Oh, wow.
You'd be forcefully stabilizing those progenitor RNAs and commanding the tissue to regenerate.
That's incredible.
Or, conversely, for a hyperproliferative disease like psoriasis, carefully tuning the SUMO2 pathway to gently force those overactive cells to mature and settle down into a barrier.
It moves dermatology and wound care from a game of mere management to a game of molecular reprogramming. We are literally talking about rewriting the instruction manual in real time on the surface of your skin.
It is a totally different paradigm for how we view healing.
And it makes you wonder, if these tiny tags are the master controllers of biological aging or regeneration in our skin, what are they doing deep inside of us? I mean, could a hyper-targeted NEDD8 hack one day be the secret to regenerating a failing heart or repairing a damaged liver?
It's definitely possible.
If the toggle switch exists in our outer shield, it might just be the key to our internal engines as well. Something for you to ponder the next time you get a simple paper cut and watch it magically disappear. That's our deep dive for today. Thanks for exploring the microscopic universe with us.